Hello everyone and welcome to our module on signaling pathways. In other videos I talk about how hormones generate various clinical and physiologic effects, but in order to do this, hormones have to tell cells to modify what they are doing. So that's what we're going to talk about in this video, how hormones signal cells to change their activity to generate the clinical effects of the hormones. Let's start by talking about the easiest hormones to understand, and those are the ones that are lipid or fat soluble and therefore they can cross the plasma membrane. This means that they exert their effects by binding to receptors that are intracellular. The receptors for all of these hormones are found in the cytoplasm or the nucleus. Once the hormone binds to the receptor goes to the nucleus and modifies gene transcription. All of these hormones shown on the screen here are lipid soluble. Most of them are steroid hormones derived from cholesterol. And in the other videos where I talk about these hormones, I've tried to show you their structure so you can remember the ones that are fat soluble. And if you just know this short list of fat soluble hormones, it's very easy to remember for your exams that all of these hormones are going to cross the cell membrane and bind to receptors inside of the cell. None of these hormones bind on the surface. None of them transmit a signal to the cell via a complex second messenger like we'll talk about later in this video. So these are some steroid hormones shown on the screen. The sex hormones like estrogens, androgens, progesterone, these are all steroid hormones and act intracellularly. Aldosterone is also a steroid hormone, and so is cortisol. Thyroid hormone is somewhat unusual in that it's not a steroid hormone, but it is fat soluble and it can cross the plasma membrane of cells. I talk about this in other videos, but thyroid hormone comes in two forms, T3 and T4. They are both synthesized from the amino acid tyrosine and iodine, and these are the structures on the screen of T3 and T4, both lipid-soluble hormones that act intracellularly. All the lipid-soluble hormones are not soluble in plasma, therefore they need to circulate bound to a protein. And so if you know that list of lipid-soluble hormones, then you know the list of hormones that act intracellularly, and you also know the list of hormones that circulate bound to a protein. Estrogen and testosterone circulate bound to sex-binding globulin, which I talk about in other videos. Thyroid hormone circulates bound to thyroid-binding globulin, and cortisol circulates bound to corticosteroid-binding globulin, which can also bind aldosterone and progesterone. Okay, so those are the relatively easy hormones, the ones that act intracellularly and are lipid-soluble. Now we're going to talk about hormones that are not lipid-soluble. This means in order for these hormones to modify the function of a cell, they have to first bind to surface receptors, and then those surface receptors have to transmit a message inside the cell to modify gene activity. Some of these hormones use the surface receptor itself to drive cellular changes. We'll talk about two of these, tyrosine kinase receptors and JAK-STAT. Others of the extracellular hormones increase the level of a substance inside the cell. This is called a second messenger. And when that level goes up, it drives changes in gene expression. And the second messengers that are important to know are cyclic AMP, cyclic GMP, and IP3. And we'll talk about all of these in the next few slides. Let's start by talking about hormones that work through what's known as a receptor tyrosine kinase. And the prototype for this form of hormone is insulin. If you're going to only know one hormone that works through a receptor tyrosine kinase, know insulin because it's the best studied example in this class. What insulin does is it binds to a receptor that has tyrosine residues. Tyrosine is an amino acid, and there are some tyrosines sticking off of the receptor. Once insulin binds, those tyrosine residues become phosphorylated. And the phosphorylated tyrosine residues can interact with other cellular components that lead to gene transcription. This is sometimes described as autophosphorylation because the receptor phosphorylates itself. And you want to keep in mind the big picture points here. This type of system requires only the receptor. The receptor does all the work. It phosphorylates itself and transmits the message for gene transcription. That's in contrast to others we'll talk about later that require a second messenger. So like I said, the main hormone you want to know that works through a receptor tyrosine kinase is insulin, and I talk about this in detail in the videos on the pancreas endocrine functions. Also, a number of growth factors work through receptor tyrosine kinases. These include insulin-like growth factor 1, fibroblast growth factor, platelet-derived growth factor, and epidermal growth factor. And these are easy to remember because they all have GF in the name of the substance. The second signaling system we'll talk about is the JAK-STAT system. This system also involves tyrosine phosphorylation, but it works differently than the receptor tyrosine kinases. In this system, the receptor activates enzymes called Janus kinases, which are capable of phosphorylating tyrosine molecules. Therefore, they are an example of another tyrosine kinase enzyme. What they phosphorylate are proteins called STATs. STAT stands for Signal Transducer and Activator of Transcription. These are protein transcription factors. They're activated by phosphorylation and then they go to the nucleus to modify gene transcription. So there's a drawing of how this works on the screen here. Many of these receptors are receptors for cytokines. So a cytokine will bind the receptor and that will activate a JAK associated with the receptor. The JAK will go on to phosphorylate a STAT, and then the phosphorylated STAT will go to the nucleus where it will modify gene transcription. So once again, try to keep in mind the big picture here. We are working through tyrosine phosphorylation here. In this case, a separate enzyme called a JAK is phosphorylating a separate protein called a STAT. The JAK-STAT system is used as a signaling pathway for many cytokines. One of the first to be described was interferon gamma. But IL-2 and IL-6 have also been described as using this system. In addition, many substances that stimulate the bone marrow work through JAK-STAT. Erythropoietin works through this pathway, GCSF does, as does thrombopoietin. And then finally, there are two others that are just different from the rest, prolactin and growth hormone. All of these receptors share homology and probably evolved along similar lines, but they're hard to remember because they come from many different systems. But the easiest way to remember the hormones that work through JAK-STAT is to just think of cytokines and bone marrow cells. In the hematology section in the video on myeloproliferative disorders, I talk about how a JAK2 mutation is associated with many myeloproliferative disorders. And this should make sense to you because we just talked about how the JAK system is involved in the stimulation of growth of many bone marrow cells. So naturally, a mutation might lead to overproduction of bone marrow cells and therefore a myeloproliferative disorder. The JAK2 gene is a gene for a cytoplasmic tyrosine kinase. Just like all the JAK proteins are tyrosine kinases found in the cytoplasm on the cytoplasm side of the cell membrane. This particular mutation leads to increased tyrosine phosphorylation and therefore bone marrow cells are hypersensitive to cytokines. And as we just discussed, many cytokines can activate these JAK-STAT receptors. The result is more growth and longer survival of bone marrow cells, and that's what leads to the myeloproliferative disorders. So now we've talked about receptor tyrosine kinases. We've also talked about JAK-STAT systems. What I'm going to do now is talk about second messenger systems, and we'll begin by talking about cyclic AMP. So some hormones can activate the enzyme adenylyl cyclase. What this enzyme does is it takes some of the ATP, the adenosine triphosphate floating around in the cell, and it converts those molecules into a molecule of cyclic adenosine monophosphate, which is called cyclic because the phosphate group forms a little ring here. Once this happens, the level of cyclic AMP inside a cell is used as a second messenger to drive cellular changes. Hormones that use cyclic AMP as a second messenger bind to G protein-linked receptors. All of these receptors can bind guanosine nucleotides like GDP and GTP, and once a hormone binds to one of these receptors, that transmits the signal via adenylyl cyclase. To understand this better, let's look at this cartoon on the screen. On the left side of the screen, we've got a G protein coupled receptor, and here's a hormone molecule coming in to bind. On the other side of this receptor, you can find alpha, beta, and gamma subunits of the G protein coupled receptor. The alpha subunit in the inactive state binds a molecule of GDP. Once a hormone binds the receptor, however, it sheds that molecule of GDP and instead binds GTP. At this point, the alpha subunit can separate with the GTP and activate adenylyl cyclase, which is the AC here, and then that enzyme will do what we showed on the last slide and convert ATP into cyclic AMP. And this is how hormones that use cyclic AMP as a second messenger system transmit their message from outside the cell through the G protein receptor to increase the level of cyclic AMP. The cyclic AMP system is very popular in the human body. There are many cells that use this second messenger system to transmit signals through hormones. So let's go through some of those now. In the hypothalamus, cells respond to CRH and GHRH through cyclic AMP second messenger systems. Many anterior pituitary hormones work through cyclic AMP, including the ones I've shown on the screen here. PTH in the parathyroid gland works through cyclic AMP, and then there is a collection of other systems that work through cyclic AMP. This includes cells when they react to glucagon, when they react to ADH through the V2 receptor, which is the receptor that's important for retention of free water in the kidneys. Histamine receptors, the H2 type, which are found in the stomach and are associated with stomach acid secretion. These work through cyclic AMP. And then finally, HCG and MSH, which is melanocyte stimulating hormone. Cells also respond to these two hormones through cyclic AMP second messenger systems. In the video on the pituitary gland, I talk about how many pituitary hormones have the same alpha subunit. FSH, LH, TSH, and also HCG all have the same alpha subunit. And if you know this, then it's easy to remember that all of these use cyclic AMP as a second messenger system to generate their effects. MSH, I mentioned before, is melanocyte stimulating hormone, and in the video on adrenal disorders, I talk about how this hormone causes the skin to turn brown or become hyperpigmented in Cushing's disease. The reason this happens is because there is a molecule in the body called pro-opiomelanocortin. This is a precursor of ACTH, but it also is a precursor of MSH and all of its subunits. What this means is, if you look at the bottom of the screen, anytime the body wants to generate lots of ACTH, it generates lots of POMC. The POMC then goes on to become ACTH, which is fine, but it also goes on to become lots of MSH, and that's why MSH goes up when you have ACT levels being high in Cushing's disease. So if you know this relationship, then that's another easy way to remember that both ACTH and MSH use the same second messenger system, cyclic AMP. Our next second messenger we'll talk about is cyclic GMP. This shares many similarities to cyclic AMP, but it's found much less commonly throughout the body. Some hormones can activate the enzyme guanylate cyclase, which converts GTP, which is guanosine triphosphate, into cyclic GMP, or cyclic guanosine monophosphate, and the level of cyclic GMP can serve as a second messenger. There are only a couple of places in the body where cyclic GMP is important as a second messenger. The first one is with hormones released by the heart. BNP and ANP, which I talk about in the cardiology section, are released by cardiac myocytes, and they antagonize the renal angiotensin-aldosterone system. They both bind to receptors called natriuretic peptide receptors, or NPRs, and this leads to vasodilation and diuresis. And the way they function is through a cyclic GMP second messenger system. The other substance that functions through cyclic GMP is nitric oxide. I discussed this in the musculoskeletal section in the video on smooth muscle, but by way of review, nitric oxide is also called EDRF, or endothelium-derived relaxing factor, and it gets its name because it's synthesized or derived from endothelial cells, and it causes smooth muscle relaxation and vasodilation. And the way it does this is by increasing the levels and activating cyclic GMP inside of cells. So a cyclic GMP system is used only by these substances shown in this slide. If you want a way to remember them in your mind, they're all vasodilators. Nitric oxide is a famous vasodilator, and BNP and ANP antagonize the RAS system, and therefore they are also vasodilators. Our final second messenger that we'll talk about is inositol triphosphate, which is also called IP3. Some hormones can activate the enzyme phospholipase C, which converts a molecule of PIP2 to IP3. PIP2 is phosphatidylinositol biphosphate. It's basically a ring attached to a long-chain fatty acid. What phospholipase does is function as a lipase and lop off the fatty acid. This leaves behind only inositol triphosphate, which is IP3, which can serve as a second messenger. Just like the cyclic AMP system, the IP3 system works through G protein-linked receptors. A ligand can bind to the receptor, and the receptor then can activate phospholipase C, which converts PIP2 into a molecule of IP3 plus a separate molecule of DAG, which is not a second messenger, and the level of IP3 in the cell can go on to trigger cellular changes. Shown on this slide are some of the hormones that work through an IP3 system. In the hypothalamus, two hormones that come from there use an IP3 system, gonadotropin-releasing hormone and TRH. The posterior pituitary hormones oxytocin and ADH use an IP3 system. Only the V1 receptor for ADH, which leads to vasoconstriction, uses an IP3 system. The other receptor, V2, uses cyclic AMP. And an easy way to remember this is that IP3, the first letter is like a 1, and therefore the V1 receptor has an IP3 system. Histamine also has multiple subtypes. The H1 receptor, which is found in the skin and in the lungs. This is the receptor that leads to vasodilation and allergic reactions, or bronchoconstriction and allergic reactions. This H1 receptor uses an IP3 system, and once again, the 1 in the receptor is like the 1, the first letter of IP3. And then two others include angiotensin II and gastrin. They work through IP3 systems. So now that we've gone through all that, let me give you my system for remembering which receptors and which hormones use which systems. There are lots of ways to do this. You don't have to use my system, but this is what's worked for me. So the first question to ask yourself is it a lipid-soluble hormone? If the answer is yes, these are the easy hormones, the steroid hormones, thyroid hormone. They all function by binding to intracellular receptors. They have none of that complicated stuff like tyrosine kinase receptors, G protein, second messengers. None of that is involved. The next question to ask is whether the hormone is one of that small group of vasodilators, BNP, ANP, or nitric oxide. If yes, then it is one of the few that use a cyclic GMP system. The next question is the hormone insulin or a growth factor? If the answer is yes, then they're talking about a tyrosine kinase system. After that, you can ask yourself whether the signaling molecule relates to cytokines or bone marrow cells. If the answer is yes, then it's going to be a JAK-STAT system. Next, you can ask yourself if the hormone is prolactin or growth hormone. These are the two special hormones that use JAK-STAT that are not cytokines or bone marrow substances. And then if you go through this whole list and you still haven't come up with an answer for which receptor type, you're going to be dealing with cyclic AMP or IP3 as a system. It's just hard to keep straight which ones are which. Many of these cyclic AMP and IP3 hormones come from the hypothalamus. There are two hormones in the hypothalamus that use cyclic AMP and two use IP3. You can come up with any system you want for keeping these straight. The anterior pituitary also produces a number of hormones that use a cyclic AMP system. We talked about these. They include ACTH, TSH, LH, and FSH. There are two anterior pituitary hormones, though, growth hormone and prolactin, that use JAK-STAT systems. And then here are the other IP3 and cyclic AMP receptors. Remember what I told you, IP3 has a 1 for the first letter of its name, and that means that ADHV1 receptor and H1 receptors have IP3 systems. The other two IP3 hormones are gastrin and A2. Cyclic AMP works on all the 2 receptors, so the H2 receptor and the V2 receptor. And that concludes our video on signaling pathways.